Hello, I'm Dr. Malescu, and today we will have a little mini lesson on resting membrane potentials. Let's get started. So, resting membrane potential, what is it all about? Well, some tissue in our bodies are excitable tissue. Specifically, in here, we're looking at a neuron which carries out the nerve impulse. So, nervous tissue is excitable tissue. Muscle tissue is excitable tissue. They have a built-up potential energy due to the presence of electrolytes inside and outside of the cell membrane. So we have electrolytes, as you can see here. We have sodium and we have potassium. And we are going to go over the fact that a change in concentration is going to change the charge, therefore change the resting membrane potential measurement. We can go in and measure at rest any cell membrane that is excitable, meaning muscle fiber or a neuron, and determine that, give or take, it's about negative 70 millivolts. But how do we get to this negative 70 millivolts is the analysis of the day. This is what we will discuss. So now let's discuss what a membrane potential is. It is the electrical difference that is present at the resting membrane, and it's a difference between inside the cell versus outside the cell. So intracellular versus extracellular. Now, that activity for muscles is to initiate contraction. That activity for neurons is to initiate a nerve impulse, which is basically an electrical signal, but it's based on the chemical concentration change. So it's an electrochemical event. Therefore, in conclusion, I would pretty much round off by saying excitable tissue is going to be exactly the same between muscle tissue and nervous tissue in terms of understanding the concept of what resting membrane potential is. I gave you the example here of nervous tissue, specifically a neuron, but this can apply also to muscle tissue. And remember, it doesn't let through anything that's large. Large molecules do not get through this biphospholipid layer. Okay, I drew out, and you can see here the lipids facing each other because it's a hydrophobic molecule. So this biphospholipid layer is a barrier to large molecules and charged molecules. So let me repeat that. Okay, the cell membrane is a barrier to large molecules and charged molecules. So how do charged molecules get in, and how do we even get a resting membrane potential if, like I said, charged molecules can't get in or charged ions can't get in? Well, we have channels, protein channels, okay? And so what we're looking at is the sodium potassium ATPase channel right here. So this is number one. I labeled it because this is what we're going to discuss first. Okay, ions, which include positively or negatively charged atoms, are elements that include, for example, sodium, as you can see here, potassium, chloride, calcium, phosphate, and there are many other different very small ions that, in order to diffuse across the membrane, they're going to have to go through a channel. So a lot of these protein channels are pumps, and they assist in getting these electrolytes in and out of the cell. All right. So like I said, these ions are not large, right? Sodium and potassium are very small ions, but that means that they are charged. Sodium is positively charged. So it means it cannot freely pass through the membranes of the cell. Okay, whatever cell we're looking at, whether it's a muscle cell, in this case, right here, I took a cross section of this portion of the neuron cell. This portion of the neuron cell is an actual axon. An axon is an extension of the cell which carries out the nerve impulse, but it must have somewhere to start, which is the starting resting membrane potential of negative 70 millivolts. How do we get to that is where we are going to move into understanding. All right, like I said, these are charged ions, which means that they cannot freely pass through, so they're going to pass through this pump. All right, So my next point is this. The resting membrane potential. If you have to look at all the sodium around the membrane, okay? Look at all the sodium around the membrane here, okay? And most of it sits outside of the cell. You notice you've got a lot of sodium here sitting outside of the cell. Do you see a lot of sodium inside the cell? No. It's just look at the sodium molecules. These are the sodium ions, pardon me. These ions are colored in purple spheres. Now look, all you see is one here. You see a whole bunch here, okay? So if you were to look at all the sodium around the membrane, most of it sits outside of the cell, while on the other hand, the potassium right here sits on the inside of the cell. Okay, so that's an important point to understand when it comes to knowing what the resting membrane potential is in terms of where are the ions located. Okay, and how does that create that negative 70 millivolts? Okay. So the reason why we have more sodium outside and less sodium inside is because, wait for it, the sodium potassium ATPase pump, which you can see right here. That's number one on my list, and I'm going to go in this direction. I know we're supposed to go left from right, but in this image, I felt that I can just really get the point across, showing you first, the sodium-potassium pump. 3 sodiums come out, 2 sodiums come in. How does this happen? 3 sodiums are chucked out, okay? Two potassiums are chucked in. This is going to require ATP. That's why it's called the ATPase sodium-potassium pump. It's active transport, okay? So what this pump specifically does is it takes 3 sodiums, like I said, you see the arrow here, 3 sodiums out. That's why I wrote this over here so you can read it and understand that 3 sodiums, 1, 2, 3, 3 sodiums are chucked out and then 2 potassiums are chucked in. ATP is used. That's called the sodium-potassium pump. Okay, so that is the first principle in understanding how we maintain a negative membrane potential. Okay, because we're constantly pumping out 3 sodiums and then chucking in 2 potassiums. And so who does that on the cell membrane? Like I said, it could be anywhere. On this, it's the axon of this neuron. All right, and even the axon has a cell membrane because it's a continuation of this neuron, right? So that is the most important principle to understand. But wait, there's more, okay? Like I said, I want you to think about what it means for the overall charge difference between the outside of the cell and the inside of the cell. All this begins before we even talk about the pump. All right, what if this pump was not there? Even if I added up all the positive ions outside and the positive ions inside, and then we added up all the negative ions outside and the negative ions inside, the charge difference across the two would be 0. However, here's the main point I want to come across to you. Here it is. Once we pump out 3 sodiums and then 2 positive ions are coming in, potassium, there is more positive outside versus inside because it's a 3-to-2 ratio, right? Okay, so again, we're actually throwing more positive things outside than we are inside in this case. Okay, so we have 3 sodiums outside and 2 potassium inside, which means we have a slightly greater positive charge, okay, on the outside. And that's why when you're looking at this image here, we are looking at seeing a lot of positive, and that is a representation of the fact that we have way more positive charge on the outside than we have on the inside. Why? Because when we started at the beginning of this review, we noticed all these little purple spheres representing the sodium ions. Well, they're positively charged, right? So anyway, I just wanted to make sure you realize that. But here's the thing. The difference is still only about -5 millivolts. So with the sodium potassium pump, with all this sodium getting pumped out, and that's what I drew over here, this is a cross section, that's why I put CS, cross section of the axon. We can see here the sodium getting pumped out, 3:2 ratio, right? All right, even with the sodium getting pumped out, we still only have a -5 millivolt difference. Well, that's not enough to get to -70 millivolts. So how do we get to -70 millivolts? Number 2. What is number 2? It is the potassium leak channel. So what is this potassium leak channel? It's basically potassium, all right, slowly leaking. What does this even mean? Well, it means that this -5 difference charge from inside to outside, we actually need for excitable tissues to be around -70 millivolts. To get to that -70 millivolts, we have this potassium leak channel. Okay, so it's kind of like the potassium leak channel has a doorway, little doorways. Now notice I'm showing you just this leak channel, but there's, multiple leak channels and multiple sodium potassium pumps, right? Okay, so this leak channel is kind of like the door is cracked open, and it means that some of the potassium is actually going to sneak through and diffuse outside of the cell, taking its positive charge with it. So now we're getting even more potassium alongside the sodium potassium pump. So in this case, we are looking at more positivity because of the potassium slowly leaking out. Okay, so it's diffusing outside of the cell, taking its positive charge with it. Remember, the concentration change leads to a charge change, and the charge change leads to a membrane change. Okay, so a membrane potential change, I should say. All right, so what's next? Now, here's what's super important. What I have established thus far is that we know that the sodium potassium pump makes this negative environment, but it only reaches to about -5 millivolts, okay? Because we have 3:2 ratio, 3 sodiums get pumped out, all right? And 2 potassiums get pumped in. ATP is used because it's a sodium potassium ATPase pump. Great. And then to get to more negativity inside the cell to create that -70 millivolts resting membrane potential, more of that potassium has to leak out through these leaky channels, and it diffuses out, taking the charge with it, making the outside extracellular space even more positive in reference to intracellular space, so that voltage change outside of the cell is even greater than what is inside in terms of more negativity. That is a gradient. The greater the difference in gradient is going to create that -70 millivolts. All right. Now, this is what we have. It is a potential for change. What kind of change? The first scenario is that we can change the charge, right? The second scenario is that it is the potential for the concentration to change. So once the potassium leaky channel causes that concentration change, because potassium is leaking out and diffusing to outside of the membrane, that concentration change leads to a electrochemical change, meaning the charge changes. All right, so always remember the theme here. Concentration change leads to a charge change. So the concentration change of potassium going from inside to outside is going to eventually change the charge, making the inside even more negative. Okay? So in actual fact, we have changed the concentration of positive things inside and out, so it's going to affect the charge, and this is called the resting membrane potential. The resting membrane potential is only maintained so long as we have the sodium-potassium pump chucking 3 sodiums out and chucking 2 potassiums in, and as well simultaneously we have the potassium leak channel doing its job, slowly opening that doorway, leaking out through that little crack. Potassium is escaping, and this is only found in excitable tissue. Muscle and nervous tissue has the potassium leaky channel, and so more potassium leaves that membrane, creating a greater gradient and a difference in charge, right? Because like I said, a change in concentration of the ions changes the charge. I know I'm being repetitive, but that's how we learn the material, just kind of rehashing what you see in this image. Okay, so again, if this is a neuron, all I need to do is stimulate the neuron, and guess what? Positive sodium ions are going to rush in. All right, so we're going to get to that in a minute. I kind of tried to draw it out here. It's not a great job, but this is the resting membrane potential. And then if there's a stimulus, all right, that is going to change the voltage. So voltage change leads to a stimulus that causes the action potential. Okay, so like I said, a neuron is excitable. A muscle fiber is excitable so long as calcium moves in and rushes into the muscle fiber and initiates the cross-bridge cycle, which is another lesson for another day. So, like I said, this is basically the resting membrane potential, and what I would like to end with is the importance of understanding the resting membrane potential in terms of what happens when it's not at rest anymore, when you're stimulating it. Okay, so here is when I'm stimulating. If a neuron is stimulated, it's basically going to receive the action potentials via these dendrites, receiving the messages. All right, it has to be a threshold value of negative 55 millivolts, which means that at this point, sodium is rushing in, potassium is leaving, and changing the voltage from a negative 70 millivolts to a threshold value of negative 55. What that means is that this pump, okay, and this leaky channel, that is negated by the fact that the nerve impulse is potent and pumping in the sodium, diffusing across and creating this scenario. Here we look at the entire axon with a resting membrane potential of negative 70 millivolts that has a greater positive charge outside than inside. But then if the action potential travels, okay, it's sodium diffusing in, potassium leaving, creating a different environment. Now we're creating a positive environment inside the cell and a negative environment outside the cell simply because of the sodium rushing in. So now we're changing the membrane potential from a positive outside, negative inside to a negative inside, positive outside. And this spreads like a domino effect across the entire axon to reach what we call the structure axon terminal, which can either communicate with another neuron via a dendrite from the other neuron, or alternatively, a muscle fiber, which will initiate everything that happens at the neuromuscular junction, releasing a neurotransmitter and ultimately leading to muscle contraction. All right, so I think I beat this to the ground. I would like to just conclude with two things that I want to, actually three main points. Here we go. So if the take-home message has not been received, it will be received now. Three things you need to know about the resting membrane potential. The sodium-potassium pump, which is going to be at a 3:2 ratio, chucking 3 sodiums out, okay, 2 potassiums in. ATP is used. That's why it's called a sodium-potassium ATPase pump. Okay, that will keep and maintain the resting membrane potential at a more positive environment outside the cell and a more negatively charged environment inside the cell. Again, based on concentrational changes, right? Concentration of sodium is more extracellular at the resting membrane potential, um, and the potassium is higher on the inside, thus creating a negative environment intracellularly inside the cell membrane. Which, by the way, the biphospholipid layer, none of this would happen unless we had these pumps. So that's point number one. Point number two, principle number two is in order to achieve that magical negative 70 millivolts that we measure to be give or take across most nerve tissue and muscle tissue. It could be more, it could be slightly less, but we measure it generally to be negative 70 millivolts. To get to that, okay, we still need yet another pump. And this second principle is to understand the potassium leak channel. Okay, if we didn't have the potassium leak channel, there would only be a change of negative 5 millivolts. And if we analyze, like I said, what ions are charged outside and ions are charged inside minus any pumps, everything would cancel out to zero. Okay, but because of the pumps, we get to a more negative number inside. Okay, so in order to get to the negative 70, principle number two is the potassium leak channel, which is going to allow the potassium to diffuse out of the cell from inside to outside of the cell, creating what we know as to be the negative 70 millivolts. All right, so and then point number three, okay, is that the sodium leak channel right here is going to be the one that will open up and allow sodium to rush in once it gets stimulated enough, okay? And what is enough? Well, the action potential travels down an axon from a neuron and changes the voltage. The process of change in voltage is called depolarization. All right, so I will save that for another lecture where we are going to move into what an action potential is, depolarization, change in voltage, and how the action potential is actually going to spread across an axon, whether it's myelinated or unmyelinated. If it's myelinated, it crosses over these gaps called nodes of Ranvier, and it'll eventually reach the axon terminal where neurotransmitters are released, but not before a calcium channel opens because it's voltage gated and causes the synaptic vesicles inside the axon terminals to release a neurotransmitter. So that is a lesson for another day. I do hope you enjoyed this little mini lesson on resting membrane potential. Don't forget, three principles: sodium potassium pump, potassium leak pump, and then the sodium leak channel, which is going to sort of allow eventually the whole action potential and depolarization event to take place. All right, take care, and I will see you next time. Next lesson. Bye.